Good afternoon, everyone.
5 minutes before the next session. So 10:45, the next session is beginning. You don't want me to beatbox for you? We always want to have a long conversation with a coffee, and unfortunately, the time is limited. So we're gonna move on to the next session, which is about the biomechanism. All right, we have a biomechanism session, and our guest speaker for today, I'm glad to introduce Dr. Merit Maduk from Stanford University. She's a world-renowned biophysicist in the field, and I'm so glad to have her here today. Dr. Maduk, first, please come in.
Thanks for that great introduction. I'm really pleased to be here at this neuromodulation meeting. I'm learning so much. So my expertise is ion channels, and I'm happy to, so I'm an ion channel biophysicist, not an ultrasound biophysicist, but very happy to be working in this area and happy to tell you about my lab's work on it. So as you know, it is no small task to decipher the complex mechanisms of ultrasound neuromodulation. From the initial ultrasound, can I point here? There we go. Does that work? No. How do I point? Oh, but then, well, I guess I'll do this.
means if you move all the way out, you should be able to.
Oh,
have
oh,
this
got it.
gray.
Got it. So,
Do you know which direction? Sorry. There, there it is.
all right, from the initial ultrasound stimulation to the final behavioral or neurophysiological output, there are many steps. And the output can be, as you know, there are many of them. So that just adds to the complexity. At the very minimum, you have to consider what's the physical part of ultrasound that's doing this, but then what of the literally hundreds of ion channels in the human brain, which are being targeted and how, and that's not to mention exocytosis, synaptic transmission, which is a huge part of brain function. So that is complex. And then on top of that, neural circuits can integrate in nonlinear ways that are very non-intuitive. So it is a big problem. As I said, my lab's focus on this has been on the molecular cellular end, which I hope to convince you is an important part of our, to contribute to our global understanding. So we've worked on model membranes, ion channels and HEK cells, and rodent brain slices, and I'll tell you a little bit about each of those today. But before I do that, I just want to put in a plug for the value of model systems. I am not going to solve the problem of the mechanism of ultrasound neuromodulation on my own in my model systems, but I think these data are very valuable because they tell us how ultrasound interacts with these fundamental units of electrical excitability. So for one example, I've had people say to me before, oh, well, in these in vitro systems, the frequency dependence is the opposite of what we see in vivo. So are you even looking at the same thing? And I wanted to highlight this really nice paper from Kim Butz-Pauley and Steve Bacchus's lab, pointing out that these ostensible differences in frequency dependence can actually be explained can be modeled into a single coherent mechanism based on the radiation force having a mechanical effect. If you simply adjust parameters to include for the volume of stimulation and the circuit integration. So again, just my plug for the value of looking at this at all levels of biological complexity, so we can take all of the data together to understand mechanisms. So back to my data. We started with model membranes. And what's the reason for that? Well, in my lab, one of the techniques we use is called bilayer recording. And I like it because it is the system that gives you the most exquisite control over all of the experimental variables. Phospholipids spontaneously form bilayers that are, a mimic for a model membrane of a cell. So we have a chamber where we have an upper and lower chamber with a small hole between. If you paint lipids across them, they will spontaneously form a bilayer. And then you can have electrodes on either cell and measure everything. You can have completely control over the composition of the solutions of what goes into the bilayer, ion channels, proteins, et cetera. So it's a really great system. And we were able to integrate it with ultrasound fairly easily. So here's the cartoon of the system, and then the actual picture from our lab. There's the bilayer chamber that I milled myself. I was proud of that one. And the ultrasound transducer. So we had planned to put ion channels into this and study this. But before we did that, what we saw was enormous currents in response to ultrasound in a protein-free, ion channel-free membrane. This was not anticipated. These currents are they're orders of magnitude higher than we would see from a single channel. So we had to address what these were. And this was published quite a while ago, but I'm going over it because I think it fits into our current model of our thinking of the mechanism, and it's related to what we'll see in ongoing studies. So these currents are not from cavitation. They occur at multiple frequencies, and including non-cavitational frequencies. I should say, all the ultrasound I'm showing today is continuous wave. So if I don't say that, you'll just know that it's continuous wave. And these currents cannot be ionic. They are capacitive currents. We're recording under voltage clamp, which means that any ionic current would have to be positive or negative according to the reversal potential for the ions, but our currents are going in both directions. Must be a capacitive current. Biological membranes are capacitors. They're a thin layer of non-conducting grease. So the capacitance is just charge over voltage. So if you have a change in capacitance, you get change in charge versus time, which is current. So we're seeing capacitive currents. And the capacitor is, so in the bilayer, the capacitance can change. You're not changing the material, but you can change either the area or the thickness will give you a change in capacitance. And what was not intuitive to me, you calculate a very small, like sub % change in thickness will give you that level of currents. And so our interpretation, I think, is shown. Oh my goodness, I was showing you the wrong slide. There you go. Now I'm back. So anyway, capacitance, thickness, area. So our interpretation of this is the capacitance is changing, and that our bilayer initially is actually a little curved to start with, just because of the osmotic pressure in the chamber. Then the ultrasound impinges on that. The mechanical force of the ultrasound causes a change to curvature, and it actually oscillates a little bit before settling down to its resting level. And then when the ultrasound goes off, you release that pressure and it oscillates back to its new curvature. So that's the model. It's ultrasound radiation force because of the frequencies. There's a change in capacitance that would be explained by this. And we can test this actually, because this means that the bilayer is moving, the membrane is moving. So we should be able to measure that with a technique such as laser interferometry, which can measure the velocity of a moving target by measuring the timing of the return of the laser. So we did this. We took our apparatus over to this instrument. We concurrently measured velocity. We could measure, we did see the bilayer moving. Not only that, it moved with exactly the same timing as the current that we concurrently measured in response to ultrasound. So that's a pretty definitive result, saying that ultrasound radiation pressure is causing a change in curvature of the membrane, bilayer capacitance. Of course, we want to know what happens in cells. We don't think that we're going to have these big movements in cells. In cells, if they've got your cytoskeleton is tethering you down everywhere, it's going to be much stiffer. However, what we do think is that there will be local pressure on the cells, and that is bound to activate mechanically sensitive ion channels. So rather than looking them in this bilayer that's moving like crazy, we moved to a model system to ion channels in HEK cells, which are just a standard cell line for heterologous expression of ion channels. And we started, so our setup is a little different, basically the same. Ultrasound transducer below, we've got cell cultures overexpressing ion channels and doing patch clamp recording. All of these experiments are, goodness. We started by looking at piezo 1 channels, exquisitely membrane mechanosensitive ion channels, with the logic that if we're having a mechanical effect, we should see something there. Oh, this thing is, the timing of this movement is a little bit crazy. Okay, so we did see an effect of, we did show ultrasound activation of piezo channels, and we saw that very nicely in the talk yesterday across all kinds of scales of biology. What was surprising here was were in the details. When we patch clamped a cell, there we go. When we patch clamp a cell adhered to the bottom of the chamber, we actually saw no effect. We did not see any effect until we detached the cell, raised it above the surface. Then we saw very robust, strong response to ultrasound, typical classical inactivating piezo 1 currents. This was not just an effect of detaching the cell, because then if we took the detached cell and moved it back down to the ground layer, the effect went away. This is reversible, repeatable, very robust. So what that means is it can't be directly radiation force, because the radiation force should stay about the same or maybe go a little down as you bring the cell up. Can't be temperature. Temperature is going to go way down if there's any change. The only thing that correlates is streaming velocity. And I don't know if the movie's going to, oh wait, yeah, so temperature doesn't, but streaming velocity goes up as you move away from the surface. And I don't know if this movie will work. Oh yeah, you can see, we could even see that. As you move the cell up and you do the ultrasound pulse, you can see the cell moving in response to the ultrasound. So we also looked at NAV 1.2 channels. They're a little bit mechanosensitive. All channels are mechanosensitive, just like all channels are temperature sensitive. This is, a fact of living in the world, but they're much less mechanosensitive than piezo. And we saw that there was no no effect on the magnitude of the currents, and a small effect on the kinetics, but that went away when we moved it up as the temperature effect went down. So no mechanical effect on sodium channels. So that leads us to the conclusion that it's acoustic streaming, which is a result of radiation force activating piezo-1 channels. So again, you might be thinking, well, so you're not going to get acoustic streaming in a brain. So what's going on here? We do think it's relevant in tissue. Again, we saw that really nice talk yesterday from Jijun and the correlate. There is a correlate in tissue, which would be, you're not going to have streaming, but you'll have a displacement of the tissue and a strain or a pressure on the tissue. And so the hypothesis that in tissue, ultrasound-induced mechanical strain or pressure will activate mechanosensitive channels. So of course, we need to test this idea in tissue, and I think, and we have, and so far it's consistent. And again, I just want to, this is not my work, but just tie this into other pieces of data in the literature. This is from Steve Bacchus's lab, who studied ultrasound effects on retinal circuitry. And there, what they see, they can see in the retinal circuits respond very robustly to ultrasound. The cells that are, oh my goodness, yeah, the cells that are removed from the surface, they can see them moving in response to ultrasound, and they have a electrical response. The retinal ganglial cells that are on the hard surface do not move in response to ultrasound, and they also do not have an electrical effect. So again, this is all consistent with this emerging picture. So we said, okay, that's in retinal circuits, how about a brain? And now we're moving to brain slice, still in vitro, but now a connected brain slice. And what I want to say here too is that our data in our lab are consistent with this picture of cells moving, displacing, feeling that radiation force, correlating with an ultrasound response. We did a lot of experiments on hippocampal neurons in culture. We saw some responses. They were not robust. They were highly variable. These were adhered to the surface. When we move to hippocampal slices where the tissue is raised above the surface, we see robust, reproducible, really incredible responses to ultrasound. So I'm going to describe those to you right now. And they're published, but I have a little bit of unpublished data at the end of my talk. I'd like to have more, but that's science. But I want to go through this result too, because I think this is an incredible example of how ostensibly different results. Sometimes ultrasound is inhibitory, sometimes it's excitatory. Different people are using different preparations, different ultrasound. Like, what's going on? Here we are using one preparation, one ultrasound transducer, one set of ultrasound parameters, and changing one other variable, one other relatively minor variable, and we see opposite effects, inhibition and excitation. So I'm going to show that to you now. So same patch clamp setup, but now with a hippocampal slice, patching onto a hippocampal pyramidal neuron. So this is a robust response to ultrasound. I should mention we're using a 43 megahertz because the lower frequencies are not compatible with patch clamp recording. And I'm an ion channel biophysicist. This is the gold standard for characterizing ion channels. Of course, we'll need to do complementary methods to look at other frequencies. But anyway, in all of our initial studies, we saw this robust response to ultrasound. So up here, and in every case, blue is control, no ultrasound, red is ultrasound. So here are action potential firing in response to injected current, pretty typical standard approach used in hippocampal recording. When we apply the ultrasound, there's an inhibition. I'm not showing it here, but the ultrasound can be applied either before, during, middle, anytime you apply the ultrasound, you see under these conditions, you see inhibition. And these are just summary data. This is a raster plot showing different sweeps over time. A number of action potentials is always reduced during the ultrasound pulse. And summary data here, that's quite a remarkable P. So I told you we see excitation too. This is inhibition. What happens? So here's 4 traces. Now the ultrasound's being applied in the middle of the pulse. The top 2 are similar to before. You apply ultrasound, you see inhibition. In the bottom 2 traces, all Martin is doing, the scientist who's recording these, is stimulating the action potential firing with a higher current, giving it a more robust train of action potentials. And when you have faster firing rates, you always see that ultrasound activates that activity, makes it even faster. This was really surprising to me, but to people who study hippocampal neurocircuitry, it was not surprising. There's just two regimes here. In region 1, in this region, the action potential firing rate is really limited by threshold. How long does it take the ion channels to reach threshold? But as your firing gets faster and faster, firing rate becomes limited by the ability of sodium channels to recover from inactivation. Because as you fire more and more of those channels get inactivated, and they don't recover until you go negative again. And there's so much firing that they're not spending enough time in the negative range to recover. And so that's limiting. And that perfectly explains our results with ultrasound. If you make the hypothesis that ultrasound is activating a potassium channel. In this regime, if ultrasound activates a potassium channel, that keeps the membrane potential low, it's going to be harder to get to threshold and you're going to inhibit. In this regime, if ultrasound is activating a potassium channel, you're going to go back negative more, and the sodium channels are saying, yes, I can inactivate, I can fire again, and you will be excitatory. That's exactly what we see. So one effect on an ion channel explains both of these ostensibly opposite effects on excitability. And we have a lot of data supporting this hypothesis. And in fact, our hypothesis is specific that it's a potassium channel from the K2P family of ion channels. These are very mechanically sensitive. They're also temperature sensitive, but much more mechanically sensitive than all the other potassium channels. And there's a lot of data here. How is my time? My time's good. Okay, so these, unlike voltage-gated potassium channels, they don't inactivate. So you can pulse in the middle, but anywhere you pulse in the action potential of firing, you get the same effect because these are constant channels, non-inactivating. So another prediction, if you're activating a potassium channel, you should decrease the action potential rate because they're activated, they bring the action potential down sooner. That's exactly what we see every single time, whether activating or inactivating, you always see a shorter action potential. Also, if you're activating a potassium channel, you should decrease the membrane potential. Resting potential is near EK, but in normal situations, a little above EK. So if you activate more channels, it should go a little below EK. And that's what we see here. So this is looking at the resting membrane potential, add ultrasound, you get a little bit decrease in resting membrane potential. So this is everything we've looked at is completely consistent with this model. I should say there's one specific K2P inhibitor published in the literature, but it's not available either commercially or from the person who made it. So we couldn't test that. But what we did, so then I mentioned that these are highly mechanically sensitive channels. We actually can't distinguish right now whether the ultrasound effect is through temperature or mechanical. They're both consistent with our data. In the modeling of the transection, we do see a little bit of a temperature increase, which would be consistent with the temperature sensitivity of most K2P channels in the hippocampus. But we also see, remember capacitance change, bilayer mechanical. Look at this little capacitance changes are always faster than ion channel gating. This little blip here, very reproducible, that's a capacitive change. Ultrasound is inducing an on and off capacitance change. And you can integrate that current and calculate a force. It's about 1 millinewton per meter, which is well within the threshold for potassium K2P channel activation. So both things could be going on here. All right, so anyway, so everything together on our limited end, but it matches with a lot of data. The PA's talk yesterday, Kim's talk this morning, a lot of you. Ultrasound is exhibiting, the radiation force from ultrasound is exerting a pressure on the membranes, is activating mechanically sensitive channels. So that's consistent with everything we've seen right now. So what is the next step for us? I'm leaving it to you to study humans and primates, but we would like to keep connecting to these higher order studies, which is of course why we care about ultrasound neuromodulation in the end. So what we've been working very hard on the last year is to develop an in vitro preparation that has spontaneous activity. I mean, that's what brains are. They're active all the time. They're not just sitting there like a slice that we had to stimulate in order to get activity. And there are many such preparations. You have theta oscillations, you have gamma, you have beta oscillations in normal brains, and there's ways to get those in vitro. My lab hadn't done them. We can get them now. So I'm going to show you a little bit of preliminary data on that. Again, this is not ready for prime time, but since this is a friendly meeting and you want to see something new, here it is. So yeah, the goal is to characterize ultrasound in a well-connected brain slice preparation. And our preliminary data is the following. We're using neocortical theta oscillations, and we're using a new Lamb wave transducer that some of you might find very interesting. I will do my best to describe it. And I'm going to do my best to describe it. So this was Pierre Curie-Jacob's idea. He's our collaborator and electrical engineer. And he pointed out these are just 3 examples of devices made in his lab. The device consists of a piezo cylinder, so vibrations in the piezo cylinder, and it's bonded to a glass slide. Glass slides are what electrophysiologists put our samples on, thin glass, so it won't heat very much. And so the oscillations in the piezo will induce Lamb waves in the slide. So your ultrasound transducer is also your experiment, where you can put your sample on. The sample just goes directly on the transducer. No need to couple of it. It's just right there. And so and there's just a well here so that you can put solution and then perfuse the brain slice. So advantages, there's a couple of, why did he make this? There's apparently there's a way to control the spot size a little better than the other transducers we have. But a big thing is that this is very modular. Like you can take it from setup to setup with from imaging. You could image through this. You can take it to different electrophysiology rigs really easily. Lots of people have integrated these things, but this is portable. So that's the idea. Can we make it to work? Again, preliminary data here. Oh, and here's our here it is in action. There's the one of these transducers in where there's a little brain slice in there. There's a perfusion and our patch pipette. Oh, actually, I think this is an extracellular recording pipette. So we're looking at, and this we're trying a lot of preps, but what we have the most data for right now is neocortical theta oscillations. Theta oscillations are connected to sensor motory integration, and hippocampus, they're connected to navigation. But anyway, these were first characterized by Bruce McIver's lab. He's at Stanford, so he gave us advice on getting these going, and we were able to record spontaneous events in these cortical brain slices. Here's an example here of a long spontaneous theta oscillation. Here's an example of one in response to Lamb wave ultrasound. Advantage here, we can record at lower frequencies. This is 441 kilohertz. And that's this is an ultrasound artifact there, but otherwise I'm going to finish on time. Otherwise, it's going to be, otherwise the oscillation looks exactly the same. So here's the data so far. It's, again, very preliminary, but what we see a very obvious, I don't have to do stats on this, response to ultrasound stimulation. So what the protocol is here is Martin is giving 30-second sweeps where each block is 5 seconds. So he does a 5-second just recording with ultrasound, then without ultrasound, no ultrasound, ultrasound. He has a set of 60, so 30 each. And out of that, then he scored. So you remember we have spontaneous activity going on, but it's not, it's not that common. And so what he does is score if an oscillation occurred within 500 milliseconds of the initial part of the sweep, which is when the ultrasound occurs, then he scores it as a success. And so as you can see, without ultrasound, there are spontaneous activity, but much lower than with ultrasound. Ultrasound, you see much greater activity in the presence of ultrasound. And these are just repeated averages of 30 sweeps showing different pulse lengths moving from 40 to 453. As you can see, sometimes it's repeatable, but sometimes we get a drop off. So we have a lot more characterization to do on this. But I think it's an exciting preparation for parsing out what the mechanism of effect is on this native oscillation. So with that, I think I'm done. This is just reiterating our working model and ideas, and we look forward to integrating it with real brain data. And I have to acknowledge Martin Prieto is the research scientist in my lab who has driven this whole project. He's a brilliant technical experimentalist, a creative scientist. He would like a faculty position, so if anybody is offering, talk to him. Have terrific collaborators at Stanford and funding from the Brain Initiative, and I'm happy to take any questions.
Hi. The K2P hypothesis is really interesting, and it made me think of nodes of Ranvier, so in myelinated neurons. So you know how they very fast acting and they have a low refractory period, and K2P plays a part there, right?
Yeah,
So-
they have very specific localizations, yeah.
Yeah. So can you maybe speculate as to whether or not that means that myelinated axons respond better to ultrasound? And another question would be, do you think that maybe the high frequency of your experiments played a role in it being, causing these?
Yeah, so there's two questions there.
Yeah.
One is K2P localization at nodes of Ranvier. I can't remember if it's also in the axon initial segment or not. But yeah, the localization could be giving us a clue. We had discussed about with 43 megahertz. I mean, the nice thing is you can focus it. So we discussed about doing focal experiments, but actually what we would like to do eventually, and which is one reason we use the Lamb wave transducer, is to do imaging to look at this, to actually look at the strain. And we have a fluorescent strain sensor that you could simultaneously look at the electrical response as well as the strain response. And then you could see whether it's at the soma or at the axon initial segment or at the nodes of Ranvier. I don't want to speculate whether ultrasound specific to one of those or not. I think ultrasound, I mean, it's been said before, ultrasound can do anything. I actually think it can. I mean, it's so the biological complexity is so high, and the way to modulate ultrasound is so variable. I think you could probably tune it for almost anything. What was the other question? Oh, the high frequency. Yeah. I mean, we are with patch clamp recording, as wonderful as it is, as awesome as it is, you can control everything. We cannot do patch clamp recording with lower frequencies, which is another reason I should have mentioned, we wanted to go to these connected brain preps, because those recordings I was showing you are extracellular recordings, and that is compatible with lower frequencies, as you saw. So we'll be able to answer that question on frequency dependence soon, I hope. Oh, no, but there's another question.
What a great talk. Relating to sort of like in vivo data that we've seen and the role of smoothing the waveforms on excitation thresholds. I don't know if you could comment on the role of these observations of oscillations of the membrane at the onset and the offset of the
Yeah, right. So we're right next to Kim. Why aren't we smoothing our waveforms? What's wrong with us? Well, one, a lot of this was done before that data, but two, I think we should consider it. We haven't been worrying about it because, our brain slices aren't going to hear anything, but really we should move in that direction. And certainly those oscillations, right? You're getting all the frequencies at those hard edges, and that's when it's stopping. So if I was going to go back to bilayers, I'm not going to go back to bilayers because I think they were useful for heading us in the right direction for mechanical stimulation. But I would look at that in cells with those smoothened waveforms and more care to that. So thank you for that question.
All right, thank you very much. I definitely feel smarter after hearing the conversation today, especially the involvement of a potassium channel. I think it is something that I can take it as a homework. So I'd like to introduce our next speaker, Dr. Siti Nurbaya Yacob. And she will talk about interesting conversation, which is neurochemical and functional connectivity changes mediated by ultrasound stimulation in deep cortical regions.
Cool. So hi, everyone. I'm Siti, and I work with Dr. Elsa Foranyan at the University of Plymouth and at BRIC. So yeah, it's kind of moving away a little bit. It's a more macro scale effects that we're looking at, and in humans. So I'll try to follow that excellent talk just now. All right, so in our lab, we're interested in inducing reversible neuroplasticity. And we want to kind of, as Elsa mentioned in the talk yesterday, we want to induce these longer-term neuroplastic changes in order to study decision-making and learning effects. And how we want to measure this is with MRI, because that gives us the spatial resolution to look at not just the target, but also regions connected to the target, so in the whole brain. So as Elsa showed in her talk yesterday, there are these excellent studies in macaques where they've shown kind of offline effects of TUS, and you can see these changes in functional connectivity across the whole brain. So how would you do that in humans? Because a lot of the macaque protocols or the animal protocols are quite different and haven't really been shown to be safe in humans. So one of the protocols that we were quite lucky to see last year was this theta burst pattern TUS protocol. So it kind of borrows a bit from TMS. It's basically TUS applied repetitively at 5 Hertz, and it was for 80 seconds. And what Zheng and colleagues found was that when they applied this to the motor cortex, and then they kind of measured cortical excitability, they saw an increase when measured with TMS, and this lasted for at least 30 minutes after TUS. So we're like, okay, great. So we're going to use this protocol. So how are we going to measure neurochemistry and connectivity? So fMRI, you're probably a bit more familiar with. We're looking at brain connectivity during the resting state. So that's when the person's in the scanner, they're not doing anything. We just tell them to let their minds wander, but not fall asleep. It's a little bit difficult, but most of them manage to do it. These are the kinds of resting state networks that you might see, and they're quite robustly seen in the literature. The most important one at rest being the default mode network, and that has this posterior cingulate cortex at its core. The other thing that you may not be as familiar with is magnetic resonance spectroscopy. So that's how we want to measure brain neurochemistry. Basically, it allows us to measure concentrations of metabolites in the brain. So what you do with MRS, typically, we use single voxel spectroscopy. So you put a voxel. So in this case, the voxel is quite large. You put a voxel somewhere in the brain, and then you get this spectrum that separates all the metabolites in the brain according to their chemical properties. You identify the ones that you're interested in, and then you look at their concentrations. So in our case, we're interested in the major inhibitory neurotransmitter GABA and also the major excitatory neurotransmitter glutamate. The regions that we're interested in, because we're a decision-making lab, we're interested in the anterior cingulate cortex, because Elsa's shown that TUS of this region disrupts switching in the macaques. And the other region, because we're studying this at the resting state, we thought we'd include the PCC because it's quite an important node in the default mode network. So if you're not familiar with where these regions are, that's the ACC over there, and then that's the PCC, so anterior and posterior in the back of the brain. So our aims were to show that we can actually reach these deep cortical regions in humans with TUS, and that we can measure the effects with MRS and fMRI. We thought we might see a change in the concentration of GABA and glutamate at the target, and also a change of the connectivity of that target, and that maybe these changes will be correlated. And because we knew that the PCC was more important at rest than the ACC, maybe we'll see more of a change in the PCC. So that was kind of a secondary hypothesis. So our study's been pre-registered. You can have a look online over there. And we had 24 healthy adults, 14 of them were female, and they all came for 3 sessions of TUS followed by MRI. So the first session, if we didn't have their MRI scan, they'd come in just for an MRI scan. And then they did 3 sessions of TUS followed by MRI. These were at least 1 week apart and also at the same time of day because we wanted to account for effects of the circadian rhythm on levels of GABA. So during each of the sessions, they come in for TUS first. So they'll have 80 seconds of TUS. And this was either a sham where we did the same preparation, but we didn't apply the TUS. And then the other conditions were TUS of the ACC or TUS of the PCC. And then this was counterbalanced across participants. After the TUS, we rush them into the MRI scanner, get a few localizers, get their anatomical scan, and then we have their first fMRI run at about 15 minutes, usually a bit less than 15 minutes after TUS. So this was just a 5-minute resting state run. And then we have MRS of one of the regions, so either ACC or the PCC, and then MRS of the other region, so PCC or ACC. And then we ended it with another 5-minute run of resting state fMRI. So for the TUS, we used the NeuroPass CDX 500 system, and we kind of coupled it with this ultrasound gel pad, just to make sure that we get a lot of contact and lots of gel, obviously, to make sure that we get a lot of contact with the person's head and to reduce the amount of air bubbles. As I said, we used the Theta Blast protocol, and it looks kind of like this. And the intensity in free field was set to be the same for all subjects, and this was about 54.5 watts. We did our neuronavigation with BrainSight using the subject's anatomical scan. And then we did our acoustic simulations with the K-Wave toolbox, and the skull was estimated using my method for T1 to pseudo CT. So actually, I've just got this paper up on archive, and then I've put all these things on GitHub. So go and have a look and play with it. Let me know if it works. I've been told it works, so hopefully it does. And for the sham session, we just turned the system off, and then we played a sound that kind of mimicked the TUS sound through bone conduction headphones. I've put the sound here, but it's probably not going to work. Let's see what happens. How do I play it? Oh, it does work. So yeah, it's a lot softer than that, but it sounds something like that. And when we asked the subjects, all of them said they couldn't really tell whether it was TUS or sham. I guess because they weren't expecting a sham session. So the 5 subjects who were in our lab that knew there was a sham session were like, oh yeah, that's the sham. So if you don't tell your participants there's going to be a sham, it may work. But it's not so important maybe for these kinds of offline effects that you're looking at. Right, so for MRS, we use the megapress sequence because that allows you to measure GABA. We tried to optimize the size of the voxel because the MRS voxel was quite large compared to the TUS focus. But the smallest we could get it without sacrificing signal quality was about 2 by 2 by 2 centimeters cubed. We did the analysis in Gannett, and then you get these measures that are tissue corrected as well. So corrected for the amount of gray, white, and CSF in the brain. And then we did a comparison between sessions. For the resting state scans, we just did a very simple seed-based connectivity analysis with the ACC and the PCC as the seeds, and then we looked at the session differences again. So I'll just, sorry, I'll just show you some acoustic simulations. So you may have seen this yesterday in Elsa's talk. This was just kind of a sanity check for us to see that we were stimulating and also measuring in the same place. So for most of the subjects, it overlapped quite well. So you see the TUS focal field in red, and then the voxels from each, the MRS voxels from each of the sessions in blue, green, and yellow, and they overlap quite nicely. I'll just show you some simulation results, but we'll have to repeat this. So these were done at 20 watts per centimeter square, but we'll just have to repeat it again for the 54 watt size PPA. So basically we found that for the ACC, I mean, we knew that it was less deep than the PCC. It's about 10 millimeters less deep. But the amount of pressure that you get at the focus was the same for both regions. And the thing that was different was actually this focal volume. So the PCC, I guess, tended to have this more elongated focal field, so it was a little bit larger. And that also meant that there was more overlapping of the MRS voxels. So these are just some other examples. I don't know if it's very clear, but most of the subjects, I guess, fell into the top category where, we got quite good overlap with the MRS and the simulation. But sometimes I placed the voxel a little bit wrongly. So this one over here, the voxel was a little bit too close to the white matter, which isn't great. This one was a freak. I just placed it wrongly. But luckily for that session, because we're measuring here in the ACC, but we actually stimulated in the PCC. So we quite happily just dropped that one data point and it was okay. It didn't affect our results. Here, the yellow one is the sham session. So it didn't matter too much that it didn't overlap with the stimulation because there wasn't stimulation during that session. So as also showed yesterday in the ACC, we didn't see any results. But in the PCC, we saw this reduction of GABA after you stimulated the PCC, and this was compared to sham and stimulation of the ACC. So we were like, why is this happening? And the thing that stuck out the most from the simulations was the fact that the focal volume was a lot larger in the PCC. So we just did a very simple correlation in each of the sessions, and we found that it only correlated during the TUS session where you are seeing less GABA with greater focal volume. So maybe that contributes to the effect or the non-effect that we're seeing in the ACC, but we're not very sure. There was no effect on glutamate. So this is just a very kind of summary of the resting state maps at during the sham condition. So what we see is kind of in line with what's known in literature. The ACC is very highly connected with the anterior insula, whereas on the right here, you see the default mode network quite nicely. So we just did a session difference. Again, with the ACC, we didn't see anything, but TUS of the PCC increases functional productivity with the PCC. And we're also seeing this interesting thing where the first run of fMRI that happened at about 15 minutes after TUS, it was showing a different set of regions to the second run, which happened at about 45 minutes after TUS. So you can see it quite nicely here in the spider plot, where the dark blue is the first run, the light blue is the second run, and yellow is the sham. So yeah, there's kind of a temporal effect maybe, but we're not very sure. The changes between MRS and fMRI were not correlated yet, but we have some more analysis to do, so not draw any conclusions from that yet. So yeah, so we've just shown that we can reach these deep cortical regions. The effects last at least 30 minutes after stimulation measured with MRS, at least 50 minutes after stimulation measured with fMRI. And it's kind of in line with what Zheng and colleagues found, that it's an excitatory effect, so decreased inhibition, increased GABA, and also increased functional connectivity, decreased GABA. And yeah, we're not really sure why we see this lack of effect of the ACC. I think the most tantalizing theory is the state dependence, because there's no active engagement of the ACC during rest, but then the PCC is this very important node of the default mode network. We have other theories, so you can come and visit Tristan's poster later today and come and discuss it with us if you have any suggestions or anything that we might be able to look at. And I'd like to thank Elsa and Tristan, because they have been really kind of the ones that have driven this whole study, and all my collaborators, my study participants, and you guys. Thank you.
So thanks for a great talk. Maybe a comment and a question. So you talked about the posterior cingulate being more functionally connected with itself, right, after the ultrasound, right? PCC with BCC. Did I catch that correctly?
No, so it was with a range of different brain regions, so, but not exactly with itself.
Right. Okay. Okay. Increases connectivity with that targeted brain area.
Yeah.
Okay. So in thinking about what you mentioned in terms of the default mode network and its function, so what was there a subjective experience with this? What did the people make of this brain region being modulated that's involved in sense of self and introspection and things like that?
Yeah, we're not very sure about that. But yeah, don't know. It's a very good question, but we're not very sure. It could also be, yeah, maybe it might contribute to these differences we're seeing between the first and the second run. They might be, I don't know, more sleepy towards the end of the session. We're not very sure, I guess. Yeah, yeah.
No, thank you.
So thank you very much again.
Thank you.
So modulation of ACC, who knows? We could be a little more metaphysical about that. So, all right. So we're going to move on to the next presentation. This time it will be joining us by Dr. Harriet Lee Banks. She actually, unfortunately, cannot be here. So she joined us online, but will be playing the pre-recorded talk.
Hello, I'm Harriet Lee Banks from Sunnybrook Research Institute in Toronto, And today I'd like to share with you some of our exciting new work looking at applying focused ultrasound for the reduction of hypertension. Here we show that by applying transcranial focused ultrasound to the periaqueductal grey region, we can reduce blood pressure in a rodent model of hypertension. But why does treating hypertension matter? Why is it important? Well, in fact, hypertension affects 1/4 of adults worldwide, and the prevalence of the disease is growing in low-income countries. Current strategies, including anti-hypertensive drugs, are often ineffective and unsafe for many patient populations, including drug-resistant hypertension and in the context of pregnancy. Deep brain stimulation, however, has shown promise in treating drug-resistant hypertension by being applied to the periaqueductal grey region. And this really paves the way for other non-invasive alternative stimulation techniques in the treatment of hypertension, such as transcranial focused ultrasound. But before I dive into our results today, I'd like to take you on a journey back in time. I'd like to take you back to 2005, when a 61-year-old male was admitted to the John Radcliffe Hospital in Oxford. After 5 years of suffering with chronic pain in the right side of his face, this individual had suffered several failed pharmaceutical and surgical strategies, but was finally referred to the deep brain stimulation team, who after careful consultation decided to apply an electrode to the periaqueductal grey region of the brain. And remarkably, when this electrode was turned on, not only was the chronic pain cured, but the hypertension, his long-lasting drug-resistant hypertension, was also reduced. This was the first example in humans of how stimulation of the PAG region could have an effect, or could have a treatment effect on hypertension. This study was then followed up in 2010, and then again in 2011, where that specific brain region, the PAG, was distinguished in terms of its hypertensive effects versus the PVG, which had more of an effect on chronic pain. The role of the central brain region on changing blood pressure has also been investigated in rodents. Though as early as 1995, we see how cardiovascular effects can be induced by changes in the periaqueductal grey following electrical stimulation, and also through chemical strategies. This is 2005, using microinjections of L-glutamate in the rat brain, also showed the involvement of the PAG region in lowering blood pressure. And finally, the most recent study of all, 2020, we see this very exciting first result suggesting that perhaps focused ultrasound could non-invasively have a similar effect on hypertension in rats. Well, over the last year, we've been working to explore this effect further and ask a few more questions. Can transcranial focused ultrasound non-invasively reduce blood pressure in both healthy and hypertensive rodents? What's the duration of this effect? And can we use consecutive sonications to sustain it? And finally, what are the underlying mechanisms? Can we start to pick out what's happening at a cellular level in terms of this hypertensive effect that's FUS mediated? Well, in order to answer these questions, we use a stereotactic guided focused ultrasound system using a single element focused ultrasound transducer with a center frequency of 540 kilohertz. And we're using a very simple pulsing scheme here, 10 millisecond bursts fired every second at a magnitude of transmit pressure of 1.1 megapascal for a total duration of 10 minutes. You can see here that our focus is relatively large, but the center of that focus is targeted to the VL-PAG region of the rat brain. The work I'm going to show you today is a summary of 2 different cohorts. The first cohort are normotensive, healthy rats, and the second are spontaneously hypertensive rats, and they're split between these 4 treatment groups. The second important piece of equipment is how we're taking those non-invasive blood pressure measurements. And to do that, we use the CODA non-invasive blood pressure system from Kent Scientific, which, just like taking blood pressure measurements in a human, is a cuff-based system. So the top cuff is an occlusion cuff, which inflates and restricts blood flow to the rest of the tail. And as it deflates, you can see in those figures, blood is left to rush back down the bottom of the tail And from that influx of blood pressure, the system is able to calculate systolic and diastolic blood flow. As you can imagine, performing this measurement in awake rats requires really careful habituation. And we found that 5 days of training prior to treatment was necessary for the rats to become tolerant of this measurement technique. And after those 5 days of training prior to treatment, we found that tail movement was significantly reduced. The variability of blood pressure and heart rate were also reduced. Although the mean blood pressure and heart rate were not significantly changed, the variability across animals was significantly improved. So once we were sure that the rats had been well habituated to this measurement, we started on the sonication treatments. The 2 studies I'm going to show you here are performed in normotensive, healthy rats, the first of which is showing a single sonication of the VL-PAG region. So we perform blood pressure measurements 24 hours before, 2 hours after, 24 hours after, and 48 hours after treatment. And you can see that compared to our ISO-only group, that's the general anesthetic alone, isoflurane with a carrier gas of medical air, there was no significant difference after just general sedation, 2 hours after sedation. But in the context of our FUS treatment group, we were able to see a significant reduction in both systolic and diastolic blood pressure 2 hours after treatment. However, this blood pressure then returned to baseline by 24 hours. So how can we extend this effect? Well, the next treatment looked at 2 consecutive treatments spaced by 24 hours. And here we can see again, both treatments, 2 hours after each sonication, again, showed a reduction in systolic and diastolic blood pressure, which returned to baseline at 24 hours. So after a single sonication of the VL-PAG, systolic and diastolic blood pressure was reduced by 26 and 22 millimeters of mercury, and 2 consecutive treatments had a comparable effect. But how long does this effect really last? We can see it returns to baseline at 24 hours, but what happens within that time window? Well, to investigate this, we look at 6 individual rats. The traces you see there are showing those 6 rats measured every 2 hours following a single sonication. And you can see when we trace the blood pressure at this finer time increment, we can see that this blood pressure was significantly reduced up to 6 hours following a single FUS exposure of the VL-PAG. However, there was no significant change in heart rate. To confirm that this is not just an effect of damage, we applied lots of different histology analysis. The one I'm showing you here is H&E staining, which showed no signs of hemorrhage or tissue damage at 1, 7, or 14 days following treatment. So we're fairly sure we can see an effect in healthy normotensive rats, but how about hypertensive rats? To test this idea, we used a crossover study where our 2 treatment groups were either sonicated at the active control region, that's the frontal cortex, which is not reported to be associated with blood pressure regulation, or sonicated at the VL-PAG region. Now, both groups of animals use the same sonication parameters as I've shown previously. The only difference here is the focus ultrasound target location. So after 5 days of habituation, the rats received daily sonications, 10 minutes per sonication, with a 2-hour post blood pressure measurement, and then measuring for a week after. Then those groups were crossed over and were measuring for another week. So to show you those results, we were able to show that compared to our cohort of naive rats, focused ultrasound applied to the VL-PAG only showed this consistent and increasing reduction in systolic blood pressure, which persisted through the 5 days of treatments and to day 6. This same pattern was seen in the change in diastolic blood pressure. A significant reduction in diastolic blood pressure was seen up until day 6 after treatment. Looking at the magnitude of this effect, in fact, that reduction in blood pressure was sufficient to move the blood pressure from a hypertensive range into a normal healthy rat blood pressure range, and that sonication of the VL-PAG, not the frontal cortex, reduced blood pressure for 6 consecutive days. We begin to see this offline effect of blood pressure reduction on the 6th day following treatments. So what's happening? What are the underlying mechanisms here? Well, using immunohistochemistry, we were able to show that CFOS expression and UN staining were significantly different depending where we sonicated the brain. Sonicated VL-PAG regions had a 49% greater CFOS expression than the unsonicated VL-PAG, and 79% of those CFOS positive cells co-localized with UN. And we take this result as verification that we are stimulating the VL-PAG region. We see that increase in CFOS expression. We also looked at the hormone content of plasma 24 hours after sonication of the VL-PAG and found that the mean norepinephrine content in the plasma following sonication of the VL-PAG was 29% higher. And this was a greater increase than when we sonicated the active control region, where we only saw a slight increase, 5% increase in norepinephrine. Interestingly, epinephrine levels were comparable between these treatment groups. Taken together, we believe this signals an inhibitory pathway governed by a GABAergic network. So in conclusion, we've shown that blood pressure was reduced in normotensive and hypertensive rats following a single sonication of the VL-PAG, and that consecutive sonications sustained this effect and reduced hypertensive blood pressure down into the normal range. We used various methods to start to unpick the underlying mechanisms and believe these signal an inhibitory pathway through a GABAergic network. This work, we believe, has implications for developing a new treatment of hypertension that has greater safety and broader applicability for those patient populations such as drug-resistant hypertension and during pregnancy. And our future work is looking to find ways to enhance and extend this effect, including testing a whole variety of FUS parameters and using ultrasound to trigger the release of anesthetics in the brain. So many thanks to all those from the Focused Ultrasound Lab who helped to support and enable this work, as well as our funders. Thank you for listening, and I'd welcome any questions.
Thank you very much. We actually have Dr. Lee Banks available through online. Please ask a question.
Hi, great talk. So in the results, you were showing it was like over 5 weeks you did sonication. I just need some clarification as to what was going on. So if I understand it correctly, it was 1 sonication per week, and then for 6 days after the sonication, there was a result. So every week you would do 1 sonication, and then the effects would last for 6 days. Is this correct?
Sorry, these were daily sonications. So 5 consecutive sonications each day, spaced by 24 hours, and then measuring the persistence of that over the following week.
Oh, I see. So 1 week with sonications, 1 week without?
That's right, yeah.
Okay, okay, great. Thank you.
Perhaps one more question from the audience? Yeah.
Thank you. So I was wondering, again, about the focal size and the structure you're trying to target here. So how much off-target stimulation do you have in neighboring structures, and how can you ensure that not stimulation of neighboring areas is involved in the effect?
Yeah, absolutely. Excellent question. And I think that was certainly one of our current limitations with the way in which we're doing the CFOS expression analysis as well. So we are trying to use regions that we believe are unsonicated of the other hemisphere of the VL-PAG as our internal control. So we're calculating a difference in CFOS expression there in the region that we believe is sonicated versus the region outside of that focus. But certainly we're looking now at higher frequency exposures where we might be able to get a tighter focus. What's interesting in respect of some of those early DBS studies is that very closely associated region, the PVG, might be having an effect on pain response as well. So potentially that's a behavioral way to understand just how many regions are being affected as well. Lots to work on.
Thank you.
All right, well, thank you very much again from joining us from Canada.
Thank you.
All right, so I happen to be the next speaker. So we've seen many interesting talks this morning. We heard about the ion channel involvement in terms of modulating the brain function. And also we studied and looked at the ACC stimulation and its behavior effect and so on. But we've been seeing a lot of activities about modulating brain function, but what about our brain sewage system? How do we clear the brain waste or metabolic byproduct from the brain? And this motivated us to look at how transcranial ultrasound can enhance the CSF or the interstitial solute clearance and transport from the red brain. And I have to get used to this. So the central nervous system, unlike other part of the organs, it lacks dedicated lymphatic vessels that are present in other part of the organs. While it has high metabolic rates, I think it is number 3 after heart and liver. And neuronal cells have high vulnerability and has a very susceptible to the changes in extracellular environment. So therefore, it demands a very excellent waste clearance through efficient sewage system. If we kind of take it for granted that our brain function is just fine, right? But if there's something wrong with our drainage system in terms of sewage system, many bad things can happen, as you can imagine. For instance, aberrant or suboptimal clearance of the brain waste is associated with traumatic brain injury, idiopathic normal pressure hydrocephalus and stroke, dementia and Alzheimer's disease, aging, and including sleep deprivation. So this rough drawn schematics illustrate the cytoarchitecture of the brain tissue. We all know that the brain is immersed in the CSF. And the brain, I don't know, well, the brain parenchyma is protected under the skull and under the dura, and is immersed in the CSF space that are kept in subarachnoid space, SAS. And then as you can see, there's a blood vessels that are supplying the blood to the brain parenchyma underneath. Of course, there are venules. And it reaches deep into the brain regions throughout the brain. And of course, there's a blood-brain barrier as well as blood CSF barrier that kind of keep the blood stream from the rest of the brain parenchyma. One of the things I'd like to keep you noted is the CSF space, CSF that are in the SAS can actually enter into the perivascular space, PVS, through the little holes in the pia matter, including meningeal matter, through a little fenestration called stomata. And it's relatively small. It's about 20 to 30 micrometers in diameter, but it's large enough to actually have the cells, such as macrophages, can go through into the PBS. And I was always fascinated by just looking at this bird's eye view of a neurovasculature. It's from the rodents. And it's only 2 by 2 millimeter cubic block. You can actually see this large distribution of neurovasculature that are present. And just remember that CSF can reach all the way down to the capillary level through the PBS, covering entire brain volume. So let's look at there will be a metabolic byproduct and the waste product from the brain. And we have to clear these things. I mean, we are clearing it all the time anyway, but it has to be transported out from the brain. So there is a hypothesized mechanisms that governs all these. There is still ongoing studies about it. First is a passive diffusion, which will be dominant in the neuropil, which is very dense. And you probably heard about it, aquaporin 4 AQP4 channel mediated water transport, known as the glymphatic transport that you might heard about. Convective bulk flow through the CSF along the perivascular space, that is another important factor. There could be osmotic pressure differences that are driving this transport. And again, as I mentioned, there's the anatomical connectivity that actually happens through the PL and the leptomeningeal stomata. And there could be other than water ion channel transport mechanism. It's all mixed, and there's a solute exchange that are taking place between the CSF and interstitial fluid that plays all the important roles there. So one of the striking evidence that has been emerged was about the convective bulk flow and advective solute transport along the PBS. It was demonstrated by the Make and Nethergaard group at University of Rochester several years ago. So what they did was they injected a CSF tracer, in this case, green fluorescent microsphere, directly into the CSF. And they actually used a 2-photon microscope to monitor and track those microsphere movement along the PBS. And as you can see, one of the interesting things that is actually from the mice brain is that first, the PBS is not really annular that are coating the vasculature, but it actually has a little stubby wing-like structure that are shown in green there. But also the transport is more dominant in the arterial side than the venular side, which is very interesting. And the movement is also mediated by the arterial wall motion and associated with the pressure gradient that are associated with it. So questions to us is, well, among these mechanisms that we think that it governs the solute transport in the brain, what would be the one that you can target as a candidate to be affected by the ultrasound? And we think that convective bulk flow of a CSF along the PBS might be the best candidate to do so, because transcranial FUS may provide opportunities in promoting solute transport by inducing convective bulk flow through acoustic streaming. Dr. Maduk mentioned about the acoustic streaming in this morning. And acoustic streaming is the phenomena that ultrasound pressure wave that create localized fluidic flow in the media. And it has been applied in microfluidic mixing, as you can see. And also it was applied in detecting the cyst in the body as well. And it also shares the similarity with ultrasound-mediated convection-enhanced delivery. We call it CED technique. So what doctors do is, in case if the BBB doesn't really allow for the transport of the drug, what the doctors do is they inject a drug directly into the brain parenchyma. And sometimes they use ultrasounds to push it further to enhance its delivery. And this was also hypothesized and theorized by a recent report by Reghaven a couple of years ago. So we had to start from somewhere because there's not much of information to begin with how we do that. So we used the Phantom to see how the ultrasound will work. We used 200 kilohertz ultrasound. It's a little low for your consideration, but you're going to see how this will affect in the actual animal experiment. So instead of actual animal brain, we use the melamine foam, which is it's like a sponge. In the United States, they call it Mr. Clean sponge, and it will have the porosity built in. The porosity is a little bigger than actually the brain itself. It's about 150 microns. So it's not a perfect example of the brain tissue, but we wanted to start from somewhere. And to mimic the neuropil, which is very dense, we use agar gel as a target. And we immerse those targets and sponges and targets into the blue, toluidine blue dye mixed with the PBS. And then we apply the ultrasound onto the surface, and then we measure the depth penetration as well as diameter of the dye. First, we examine the effect of varying pulse durations across the fixed PRF of 1 Hertz, low intensity of 0.5 watts per centimeter square ISPTA, and we apply it for 10 minutes. And as you can see, including the continuous wave, by the way. And we found the use of 100 millisecond created the largest depth. So then in the next experiment, we used the 100 millisecond pulse duration, and we varied the duty cycle between 10 to 50%. And of course, at fixed ISPTA. And we found 10% duty cycle was the best. Interesting to see that there's nothing happens on the agar gel, which is, we believe it is passively, is just governed by the passive diffusion motion. So motivated by this in vitro finding, we decided to move on to the animal study. So we use the two CSF tracers in this case. One is a medium molecular weight Texas red ovalbumin that has about 45 kilodalton. We picked that because it has the size relevance to the A beta, amyloid beta oligomer that are associated with the AD, which is about 8 to 70 kilodalton. And we decided to give a large molecular weight, 2000 kilodalton dextran particle. And we applied that through the cisterna magna so they can be considered as a CSF tracer. And then we apply the FUS through both from the top to bottom, from bottom to up in two different cases. And this is the result. Compared to the one has not been sonicated, which is shown on the left side, as you can see, both the dorsal and ventral direction of the sonication enhance the tracer distribution, especially the part of the red ovalbumin, especially around the ventricle area. On the other hand, in terms of large molecular fixed dextran, we didn't really see that much of change. But it's also important to say that this transport happens right at the sonicated plane. We ran a numerical simulation as anticipated in the red, 200 kilohertz. It's pretty big, and it created a bit of a reverberation, as you can see. And it was interesting to see that there is a little local minima that happens around the ventricles that actually happen to coincided with the enhanced solute transport. So it is something to do with it. We also estimated the thermal elevation, and it was really low intensity, so it didn't really have any thermal effect. We just checked for the method itself. The itself is safe, and we wanted to make sure it does not disrupt the BBB, which was the case. And the later part, we decide to now move on to whether the sonication can enhance intracortically injected solute tracers. So everything else stayed the same. We used 100 millisecond pulse duration at the 10% duty cycle, but only the difference is now the tracer, both ovalbumin and the fixed dextran, was injected directly into the unilateral side of the red, and we applied the sonication to the site. And the right side shows the fixity distribution with the sonication and then without the sonication. And the left one shows the ovalbumin distribution. As you can appreciate, the sonication increased the transport of both solute tracers in both directions away from the sonicated site. And slice by slice comparison of the tracer distribution volume also confirmed the sonication enhanced the transport of both tracers with slight bias toward the caudal side of the brain. We wanted to make sure that the sonication itself does not elicit a tissue level brain stimulation. So we measured the EEG from the animal just to make sure that we believe there could be some excitatory effect that might be associated with the sonication itself, but we just wanted to see if there's any tissue level responses, but it was basically the same. So at the given sonication parameter, we don't believe that there's the tangible brain excitation that are associated. Dextran that we used here as a tracer is a long chain molecule, which is 2000 kilodaltons. So we just want to make sure that the sonication didn't really alter anything. So we compared the volume, we performed the electrophoresis of those samples and make sure that they are the same size. So this is the summary. Ultrasound can non-invasively enhance the transport of the CSF and interstitial solutes with a mechanism likely to be associated with the acoustic streaming. There are a lot of things to do in the future. First, we wanted to measure its impact on solute clearance. So we have to see whether it actually clears the solute outside of the brain. So to do so, we wanted to see if there is any solute transport out from the brain and clearance to the deep cervical lymph nodes. That's one of the things we like to do. And of course, we love to perform in vivo imaging, like a 2-photon microscope, to monitor the trace distribution, because all the experiment that we have done was under the anesthesia, and anesthesia is known to alter the solute transport in the brain. And of course, we have to optimize the parameter. We only use the 200 kilohertz, and we have a limited scope of parameters. So we wanted to make sure that we are getting the sweet spot. And the numerical simulation was coveted to see. Some people ask us to, what about, why can't you simulate the particle movement in the space? But it's very challenging because it's very hard to model the brain vasculature itself, including the PBS. And of course, we would like to apply the technique to promote actual A-beta oligomers, and perhaps promote the clearance of lactate that are associated with Alzheimer's disease or to the sleep deprivation. And of course, somebody asked us, what about the brain stimulation effect itself can affect the brain lymphatic clearance? So we don't know that yet, so we want to study that. So thank you very much for today's session, and this concludes my presentation. I'd like to thank my funders and the staff for their dedication. Thank you. All right, before we take a lunch break, we'd like to have a question from the audience. No question? Yes.
Maybe just a general question, really, but what was the, I guess, inspiration for you to explore this with ultrasound? Inspiration for you to explore this with ultrasound. So where did the idea come from? And
well, that is a really good question. So we came up with potential mechanisms that are associated with the solute transport and the clearance, and probably the convection and acoustic streaming effect is something that we can do. Of course, there are other possibilities. We can perhaps modulate the lymphatic transport and so on, or perhaps using BBB disruption as the tools to enhance the solute. But we wanted to start somewhere, so that's how we got started.
So if I understood correctly, you came up with the 1 Hertz frequency based on the phantom work, right? Which is very interesting because I think that also 1 Hertz slow oscillations during sleep have been associated with movement of cerebrospinal fluid, and this has been suggested to move sewage out of the brain during deep sleep. It's one of the functions of sleep. So might be coincidence, right? That this 1 Hertz stimulation has physical properties also that work best, or because it was not based on the brain stimulation effect that you choose it, right? But would you test different frequencies, like 1 Hertz for the slower and faster frequencies also in the brain?
That is excellent comment. So one of the things I like to emphasize is, well, 1 Hertz for humans is very different from perhaps for the rats. So because a lot of this convective motion that are associated with the arterial pulsation is a cardiac driven. So all the experiment data that we have acquired is from the rat. They have a really higher rate of heart rate than humans. So maybe there is something in there. And but one of the studies, the result that I have shown is the in vitro study. And I do not know how exactly it will translate out to the in vivo study. So it's something that we'd like to keep in mind on. Thank you for the comment. All right, so we are really relatively on time, 12:07, and I hope you will enjoy your lunch. Thank you very much.
Thank you.